Hello, everyone. It's my pleasure to be able to participate in this lecture series with the topic of transcriptomics. But first, to introduce myself, my name is Tuuli Lappalainen. I'm a professor at KTH Royal Institute of Technology in Sweden, also a director of the genomics platform of SciLife Lab and the National Genomics Infrastructure here. And I'm also a senior associate faculty member at the New York Genome Center, where I had a lab for a long time. So that's a long and complicated CV, but it's a pleasure to be talking to you over Zoom now. So before I get into the actual science, I want to say a couple of words about the person who we want to remember with this lecture series, and that's Casey Brown. So Casey was a good colleague and friend of mine. We worked together, particularly in the context of the GTEx consortium for many years, and he was really a brilliant scientist and a really fun, wonderful guy, and we all miss him a lot. Here's a couple of pictures of that I found of me and Casey with many others over the years from Cold Spring Harbor, from the Biology Genomes meeting, where we used to hang out a lot, and then also from a GTEx consortium meeting evening event. So it's been a real loss to the community when he passed, but I'm happy that we are able to come together like this and honor his memory by talking about some of the topics that he wanted and providing some educational content. So I was asked to talk about transcriptomics, and that's, of course, one of my favorite molecules, or one of my favorite omes in this world. And I just want to go really kind of like into the basics in the very beginning to maybe those of you who were asleep in high school biology classes, and just refer to the central dogma, which basically talks about how the information encoded in the DNA, in the genome, is transferred to molecular functions in the cell, driven particularly by proteins. And RNA, or the transcriptome, has, of course, a key role here in the middle, being the kind of a way how the data encoded in DNA is copied into another nucleic acid, but then it actually leaves the genome, unlike, for example, epigenomic marks that are still kind of stuck in the DNA. But RNA leaves the genome and then is encoded in the protein. And as such, RNA really has this unique role of data readout and transfer, which, in my opinion, makes it the most fascinating molecule or molecule or sort of type in biology. RNA also has molecular functions of its own, but we'll get back to that a little bit later. Another sort of like a very basic thing to get everyone on the same page is just that when we talk about transcription of genes and how that kind of like what happens there, that is kind of regulated by a whole bunch of regulatory elements in the genome that drive transcription into pre-mRNA. And then those transcripts are then processed so that you have basically kind of added things in the 5' and 3' ends to prevent the RNA from sort of like decaying. And then very importantly, interns are excised, and then exons are spliced together into a mature mRNA molecule. And there is, of course, variation in that about which exons actually get spliced together and how, creating further diversity of transcripts from a single gene. A very large part of the genome is actually transcribed, has that type of a molecular function. This includes just about 2% of protein coding genes, so basically the exonic parts of the genome. But then introns are also there transcribed, and then even though they're cut out, that's part of the sort of the transcriptome slice of the genome, and a huge part of the entire human genome sequence. And then there are also some other sort of miscellaneous elements that are sometimes transcribed at some level. And altogether, if we think about sort of the functional elements of the genome, or how do you even define function, a very large part of the genome has some degree of biochemical function. Sometimes it's transcription, sometimes it's, for example, protein binding, but really transcription forms a major part of these things. But not everything that is transcribed is actually sort of functional in the sense that it affects phenotypes that are actually somehow relevant for organisms. Depending a little bit on how you count, it's maybe about 8% of the genome that is functional in the sense that it has phenotypic importance. We can measure that by sort of like analyzing how, for example, genetic perturbations, genetic variants, or sort of artificial perturbations affect phenotypes, or looking at evolutionary conservation. But in general, there is the sort of, well, the take-home message here is that there is a lot of biochemical activity. Much of that is transcription in the genome. Only a subset of that is truly functional and really important for downstream function. Another take-home message here is, and this is probably not news to any of you, is that really that protein coding slice of the genome is very small, even though a lot of our work focuses on that for very good reasons. This is kind of diving a little bit deeper into that aspect of transcription and the small slice of protein coding genes. So this is actually a very recent paper that just came out of the most recent annotation of both the human and the mouse genomes, and the genes in them. Here I'm showing just the human part. And there's a whole bunch of numbers and things here. I want to kind of draw your attention to a couple of things. So firstly, gene annotations are kind of structured into different classes of genes. And there's the protein coding genes, the sort of like they establish the nice sort of clearest part of things. But then there's also long non-coding RNAs, pseudogenes, small RNAs, and immunoglobulin and other genes. And here the protein coding genes, we're talking about 20,000 protein coding genes. When the human genome was first sequenced, or kind of like back in the early days, people thought that there's actually many more of these. But no, it's about 20,000. The annotation has been quite stable for about 20 years now. There are small changes. As you can see here, the numbers have changed a little bit between the older and the newer versions of gene annotations, but not huge and dramatic changes. And then, of course, here, as I already mentioned briefly, from a single gene, you typically have multiple transcripts, so different combinations of exons and introns and elements of that gene that create this sort of additional functional diversity. Another very important class and a large class of annotated genes are long non-coding RNAs. So these genes don't code for a protein. They are transcribed. They are sort of, sort of structured units, not just some sort of like random transcription. These are annotated genes. And this is an increasingly well annotated and characterized class. It has been growing in recent years. You can see here the number of annotated genes and transcripts has increased substantially. It's now it's quite a bit higher than the number of protein coding genes. And some of these genes have really important functional roles, like even that kind of if something goes wrong with some of these genes, it can be lethal. But not all of them are necessarily as important for function as protein coding genes. It's a little bit more hazy, and there's a lot of work ongoing in understanding this category of genes fully. I'm not going to get deep into the other genes, just mention that the small RNA genes here, so these includes many sort of smaller transcribed genes that often have key molecular roles in transcriptome regulation and function. So, for example, the sort of smaller genes that regulate, for example, splicing, translation. We're talking about ribosomal genes, small RNA genes, etc. They are included in this class, and probably actually include some of the most important genes in the human genome in some sense, that they really drive very old key molecular functions. So another sort of clear effect of the transcriptome and transcription is that it's cell type and context specific. And I'll get back to this a little bit later in more detail. But the cell type specific activity of gene expression is a key driver in the sort of process of how the same genome across all of our cells, more or less, produces different cell types. I mean, your liver cells and your neuron cells are very different. They do different things, and that's partially due to sort of molecular processes that activate different genes, and there are very substantial differences in expression between cell types. And there's both sort of like a cause and effect here. So transcription of key regulator genes drives further differences between cell types that are then sort of reflected in the transcriptome space. And the same is true also in a more sort of subtle and more dynamic processes of context-specific cellular responses. So when you have an environmental change, how does your body actually respond to that? And those processes are also very much driven and also reflected in the transcriptome. For example, like growth factor, another receptor, can receive sort of signals from the outside that are then basically activating transcription and translation that then drives cellular responses. A key feature when we think about where does the transcriptome actually fall in the sort of like the whole mess of like different omes and different phenotypes and etc. So think that one can kind of like think about different types of omics structured in a little bit of a hierarchy. So epigenome, for example, enhancer activity or transcription factor binding is the sort of regulatory processes of the genome. These are the stuff that is still stuck in the DNA. And then transcriptome, as we discussed, is kind of the first molecule that leaves the genome, translation into proteome. You can think about different types of cellular traits that result from the different sort of transcriptome proteome processes, for instance, cell morphology or ion transport, those kinds of things. Then if we move further into more complex, more comprehensive functions, tissue functions, physiology, disease. And all of these processes, from epigenome to transcriptome to whether you have what's your cholesterol level, these are all phenotypes. These are all traits, even though they may be molecular traits that we measure with the same tools that we use to measure the genome sequence itself. These are phenotypes. They are correlated and/or causally linked to each other in very complicated ways. They respond to the environment. They can vary between individuals, partially due to genetic factors and partially due to environmental factors. And it's very important to sort of understand the distinction between something like the genome, or I'm talking about the germline genome that you inherit from your parents. It doesn't change in response to phenotypes. It is kind of like at the bottom of the causality chain. But all these other things, they are dynamic processes with an ability to respond to things and various types of sort of, even though you can structure things with a little bit of hierarchy here, like whether you have asthma can affect and will actually affect, for example, your gene expression levels, your transcriptome, because your cells are responding to the disease state, for example. So there is kind of like, it's not sort of like a simple causal pathway from top to bottom here. Everything can affect everything, but the genome is different. The germline genome is stable. Exceptions apply, but especially in cancer. But that's the general rule when we're talking about heritable genetic effects, which I think you will discuss later in this lecture series, and traits such as molecular traits of the transcriptome. So in the next few slides, I want to talk a little bit more about how do we actually analyze the transcriptome, moving away from these sort of fundamental principles of what the transcriptome is and what it does. So these days, we measure the transcriptome with RNA sequencing, and I'll give you a few slides here about some of the few different key technologies here. So about, I guess we're now talking about 20 years ago or so, the development of Illumina sequencing technologies, very, very highly scalable ways to sequence short reads of nucleic acids, really transformed this field. And the way these things work is that, I mean, obviously, first you need a sample of interest. You need some sort of cells or other biomaterials. You extract RNA from there, and then you can do, if you want, some degree of target enrichment. So what is very common in RNA sequencing is that if you're interested in primarily on the protein coding genes and partially some long non-coding RNAs, you enriched for poly-A molecules. So this is protein coding genes, and some non-coding RNAs have a poly-A tail attached to their 3' end. And if you enrich for those, you basically kind of like it allows you to sequence more of that and less of the other stuff that is less well characterized. So this is very common. Then to kind of prepare those molecules into something that you can actually load on the sequencing machine, you typically fragment these molecules that can sometimes be quite long. I mean, we're talking about thousands of base pairs in terms of typical mRNA genes. You reverse transcribe them. So we with Illumina technologies, we don't sequence the RNA itself. We sequence cDNA. So that is basically being kind of with enzymes that are kind of that have evolved for this purpose. You turn the RNA back to DNA. Called cDNA, and then you basically ligate adapters and stuff to the ends that allows the sequencing machine to sort of like catch these fragments and read them, and then you amplify them with PCR. So then this cDNA library is then pipetted into the sequencing machine, and what you get out from that is sequencing reads. So in a typical Illumina RNA sequencing type of a thing, and we do talk about RNA sequencing even though it's strictly speaking cDNA sequencing, you end up getting those kinds of like adapter kind of bits and pieces in the ends, and then in the middle on both sides you have the sequence that you're actually interested in, the sequence of those cDNA molecules. And then in a paired end type of a sequencing situation where you have from a single fragment you sequence them from both ends, you may have a little gap in the middle of an unsequenced part, but basically you have two reads reading different ends of the same molecule. And then to analyze these data to figure out what did you actually sequence, so what are these things that came out of the machine, you map these reads to either to a transcriptome or a genome reference. So you can take the human genome reference, and with some methods you take a reference library of known human transcripts. And you basically align those sequencing reads that came out of your sample into these references, and that allows you to identify that it's like, okay, so here's a gene that has been annotated, it's been identified before, defined, and here I have these many of the reads in my sample map into those positions, and that allows you to basically estimate what is the expression level of that gene in your particular sample. And then there's a whole bunch of downstream analysis that this allows, which we'll return to in a bit. So that's the story of standard short read sequencing, and this is probably like, I don't know, over 90% to probably over 95% of RNA sequencing that is happening out there is of this type. And to talk a little bit more about kind of the without getting into very much into the details, I described the transcriptome genome mapping part and the analysis part at a very kind of high level. But what actually happens is quite complicated, and there is sort of like, I mean, many careers of brilliant scientists have gone into developing very, very sophisticated toolkits to process the reads that come out of the machine, kind of like make sure that everything is as it should, and can prepare the sequences, polish them a little, get rid of those adapter bits, for example, that is kind of like the technical parts in the reads, and then to align the reads into the genome or into the transcriptome. That's a very, very data intensive process. There's a lot of data here. It requires very efficient algorithms. And then you kind of like sort out the results to kind of clean up some artifacts that may have occurred, to actually count what does overlap those gene sequences, to quantify the gene expression, and then to process the outputs into QC reports that allow you to understand if the data that you got is actually of good and consistent quality. So we're not going to discuss these things in details, but it's important to understand that there is a whole sort of universe of tools out there. But luckily, there is also pretty good pipelines that have been constructed to sort of help people to navigate that universe. You don't really need to develop these things from the scratch. But so that was the short read story. Another very important sequencing technology when it comes to transcriptome analysis, and actually also genome analysis, but now we're talking about the transcriptome, is long read sequencing. So in short read sequencing, as we discussed, you get your RNA reads, but you then fragment them and you sequence, basically, we're talking about 75 or 100 or 120 or so base pairs with each read. And as I mentioned before, the actual transcript molecules are often thousands of base pairs long. So chopping up those actual molecules of the transcriptome that are really kind of like the functional, like the real biomolecules of the transcriptome into small bits, because that's how our sequences work, that's a little bit suboptimal. So over many years, and now increasingly so, people have been pursuing long read sequencing approaches, where you can basically take your RNA, and sometimes actually with Oxford Nanopore technologies, you can sequence the RNA directly without cDNA, but often it's still cDNA. You take those molecules, and then you basically just sequence the entire molecule. We're not getting deep into sequencing technologies here, but this is a growing field, and it is something that is very important for transcriptome analysis, particularly when we're talking about those complex intron-exon structures, and basically the fact that from a single gene you often have multiple transcripts, and they can differ from each other in complex ways that is not always easy to figure out from short read sequencing data that is basically choppy. And from long read sequencing, you get a much better view into not just the kind of quantitative variation in gene expression levels, but also the qualitative differences of being able to look at the actual transcripts that really the transcriptome consists of, and that are the real molecules that go and do their jobs in the cells. Another very, very hot technology in the space of transcriptomics is single cell RNA sequencing. And I'm sure you have all heard about single cell RNA-seq many times, but as a very brief introduction, how this works is that when you have your biospecimen, your tissue sample or cell sample or something, you don't just go and sort of like extract RNA out of that. You actually disassociate the cells, unless you're already looking at something like blood cells that are already a suspension. And then you basically seek to analyze those cells individually. You can do sort of different types of enrichment of target cells if you're interested, for example, in a particular subset of the cells in your sample. But you basically use different approaches to do single cell capture, and to be able to sort of like basically The most common technology for this is provided by 10x Genomics, and the way 10x works is that you basically have a device, a microfluidic device that combines your cell sample, kind of your cells come in here, with droplets that contain beads that have specific barcodes, so that in each droplet, in this kind of partitioning oil, you have those beads that carry specific barcodes, and then a single cell. And so then what happens when you actually do sequencing here, and there are also other ways of sort of achieving a similar type of a situation that don't necessarily require the same type of microfluidics, is that the sequencing reads are in addition to having the actual sort of reads or the sequence of the RNA of interest, and the kind of like adapters, PCR handles, those kinds of sort of technical parts, they also have a barcode that tell you that this read and that read and this third read, etc., came from the same cell. And so then using computational tools, you can take that entire library of sequencing data and partition that into according to the cell of origin of the different reads. And this has been very revolutionary technology in many ways, and we'll get back to the applications in a little bit. But so let's discuss a little bit when we do these types of sequencing experiments, and then we kind of like start to look at the data. What are some of the properties of those data? So the first question could be that, like, so you had samples of a particular cell type, let's say, and let's say that we're just kind of like doing bulk sequencing here. It's a little bit easier to think than the single cell situation. But so you have characterized gene expression in, let's say, monocytes. And then you could ask that, so what is the distribution of expression levels of genes, like of these 20,000 annotated protein coding genes in monocytes? What would it look like? Unfortunately, this is a Zoom lecture, so we can't really do sort of raised hands, etc. here. But the correct answer here is the type of a distribution here in the middle. So basically, most genes are very, very lowly expressed in a given tissue or cell type. About half of protein coding genes are basically not expressed at all, or only at very, very teeny tiny trace levels. And then there is a small number of genes that are expressed at pretty high levels. And those, of course, which genes are expressed in which cell type, that varies, and that particularly defines the cell type. But in general, there is the general pattern is the same, that there is a relatively small number of very highly expressed genes, and then sort of like an exponential decay and a lot of very lowly expressed genes. One noteworthy thing is that of course, sometimes those very highly expressed genes, the small number of them, are like functionally really, really important. I mean, if you look at like beta cells of the pancreas, insulin is very, very highly expressed, and that makes sense, and that's good. But it's not always so that highly expressed genes are functionally more important than lowly expressed genes. It depends on the gene type and what is their actual biological function. Some genes that are more sort of like collagen or actinin or something that are a little bit sort of like structural components of the cell needs a lot of them. Whereas some really key molecules that, for example, regulate cellular functions, and for example, transcription factors that regulate how genes are activated, those are typically present in, or kind of like needed only in very small amounts. But let's flip the problem a little bit, and let's say that, I mean, you're not just interested in sort of characterizing what are the genes expressed in monocytes, but you have measured your collected monocytes, let's say in 100 individuals. And you're interested in like looking at, okay, so here I have a given gene, and what is the distribution of that gene's expression across individuals. Now I actually just noticed that I have a mistake here on the y-axis. This should say samples or individuals. So now here we're looking at a single gene and what is its expression across individuals. And here the distribution is different. It's more or less, this is my very bad attempt to try to draw a normal distribution. Because again, if we go back to the earlier point that I made about expression being a molecular trait. So expression is a quantitative trait. It's a little bit like height or cholesterol level or something. And those kinds of traits are typically normally distributed in a population. So there's a lot of individuals that have sort of like intermediate levels of expression of a particular gene in a particular tissue. And then there are some that are higher and some that are lower, but it's kind of like a bell curve here. And some genes are more variable than others. Some genes are quite sort of like tightly regulated. It's quite specific what kinds of amounts of that gene is needed for a functional cell and a functional organism, and you don't deviate much. And some genes are a little bit sort of all over the place, and that is related to the sort of different functional properties of those genes and the evolutionary constraint, the kind of how important a given gene is. But so moving on, so those are sort of like the sort of basic sort of distributions, how one should think about like how is gene expression distributed across genes and across individuals. But in practice, when you actually get your sequencing data out of the sequencer and you quantify gene expression and maybe splicing different types of things, you have your big matrix of like your samples and the different genes or transcripts and their expression levels. A very, very important thing in analyzing these types of data is basically first start exploring the data a little bit. Basically visualizing the data, including with sort of unsupervised methods where you don't really kind of like, you let the data guide you, you let the data tell you what are the patterns of variation and patterns of differences in expression that are there. So I'm here showing just an example of a few plots. So this is the principal components analysis, the plot of the first two principal components of gene expression of a few samples. I think I've forgotten the figure reference from this slide. Apologies about that. But so here you can see that like, okay, so there's two groups and maybe they're a little bit different, but not very much. You can also use a UMAP that is a little bit different, somewhat a more controversial way of doing this sort of unsupervised clustering of samples or cells. Principal component analysis is perhaps a little bit more interpretable. But in any case, these types of plots allow you to see if there are any kind of like big clusters and if or kind of like major differences and how that correlates with different properties of the data that may be known to you. For example, if there are two different groups or if there are some technical factors. We'll get back to that in a second. Another way to explore or kind of like start to look at a little bit what's going on in your data is differential expression analysis, and we'll return to that in a bit. Where you like if you're like for example in this situation you have two groups and you may want to ask what are the genes that are differentially expressed between these two groups? What is higher? What is lower in which group? And then another and a common way to visualize the differential expression analysis is this kind of what is called a volcano plot, where you have the significance of the differential expression on the y-axis and the fold change, so the magnitude of the difference on the x-axis. And another common way to visualize things is a heat map, where you basically have your samples here, often in columns and different genes in rows, and then you sort of like mark what's the expression level of each of these genes across the different samples, and then you can sort of see a little bit do your samples cluster in a particular way, and what are the genes that may be driving some of this, and are those genes sort of like very, very clearly sort of like distinguishing some of the different samples, or are they more variable? So just a couple of words about differential expression analysis. I'm not getting deeper into the statistical methods in any of these things, just giving you a very high level idea. But basically, this is the analysis where you look at your, for example, your two conditions of samples, and you ask which are the genes that may be differentially expressed. So here each dot is a sample, and then you would be looking at a single gene and sort of asking what's the expression level between these two conditions. It can give you an idea of what kinds of molecular processes may be, for example, associated to disease if you're talking about cases or controls. Or for example, cells with or without a drug, etc. And there are very well established methods for doing this type of analysis. One needs to control for various sort of properties in the data, but there are good methods for this. When it comes to interpretation of differential expression results, it can be a little bit complicated because, I mean, when you, for example, look at cases and controls of a particular disease, some of the differences in expression that you may see may be actually the causes of the disease, that this is kind of like really the gene that kind of like something goes wrong and that's why you have the disease. But it can also be a consequence of the disease that kind of like, I mean, something is wrong. I mean, you have a disease, so your physiology is a little bit of something is a little messed up, and that actually ends up sort of like having a feedback loop that affects the expression of a bunch of diseases, and it's a bunch of genes, and it's not always easy to tell the difference between these things. And then it can also be due to a confounder of sorts, something else that is correlated to the disease status. So these are useful analyses, but sometimes a little bit tricky to interpret what it means. Another quite common type of analysis to do with gene expression data is co-expression analysis. And that's kind of basically a fancy word for saying that we're looking at correlation of expression levels between genes. So here's an example here in these scatter plots. So here each dot is an individual, and you would be looking at kind of correlation of one gene versus the other. And some genes are highly correlated, some genes less. And then you can basically kind of construct a network out of these things so that you would have sort of strong ties between the highly correlated genes, saying that like, okay, maybe there is some sort of relationship between the two. Maybe one of them regulates the other, or maybe they're just part of a same biological pathway. And then one can sort of slice these types of networks and these data into different ways, kind of like asking that, like, okay, well, do we have these sort of like sets of genes that seem to be co-expressed, so highly correlated? What are those genes? Are they actually somehow part of similar pathways, etc.? One complication of these types of analyses is that these types of correlations can also quite easily arise from different sort of more like kind of technical confounding factors that we'll talk about in a sec. And there is often quite a lot of correlation, so that you have sort of like a big hairball of correlated genes. So all these analyses are quite common. It's again kind of the fact that it's not so easy to infer a causality and really sort of understand what are truly the biological relationship between these genes. Sometimes it makes it a little bit challenging. I don't want to sound too negative here. I just want to give a word of warning. Single cell RNA-seq analysis methods, I'm really not going to get into in any level of detail, but I just want to highlight kind of like a couple of sort of applications that are particularly compelling and popular when it comes to single cell analysis, that are not really that are less easy to do with bulk samples. So of course, in a typical tissue sample, you have multiple cell types that the tissue consists of, and just kind of analyzing what are those cell types that are present in a particular tissue, sort of building these sort of atlases of the cell type composition of human tissues, has been an extremely important scientific endeavor of the past 10, 15 years or so. And now this is getting actually to a relatively good level with human cell atlas. One can also study with single cell analysis better the sort of the dynamic processes of cellular molecular processes. So for example, if we talk about processes like cell differentiation or response to some stimuli, the cells are related to each other in ways that are not just sort of like different cell clusters, but like some cells give rise to others and then they may bifurcate, etc. And there are methods to kind of capture these types of processes from single cell data that have been very useful, particularly in let's say developmental biology and such. And then also when it comes to molecular biology and understanding, for example, the processes of transcription and how that is regulated and kind of like what actually happens, having single cell resolution is extremely useful because these are sophisticated dynamic processes with often sort of like very fine scales. And in a bulk sample, you would be basically sort of like just averaging over a whole bunch of cells and not necessarily being able to distinguish some of the more sort of sophisticated processes. The key challenge when it comes to single cell analysis is that the data is very, very sparse. So from any given cell, you typically quantify some thousands of genes, not at all the genes that are expressed in that particular cell in real biology. This is due to two factors. I mean, firstly, especially for somewhat lower expressed genes, like maybe there's actually not super many RNA molecules in the cell, and the process of single cell sequencing, basically you just kind of like didn't capture those. And then also because of sequencing costs, it is typically not feasible to really sequence every single sort of molecule that you actually captured from a particular cell, but that there's a little bit of a trade-off of doing enough, but for a feasible cost so that you can actually get data from a lot of cells. So the fact that the expression level of most genes in any given cell is 0, you have exactly 0 reads. And then sometimes you have 1 read, and if you're very lucky, you have 2 reads or more. That creates very specific analytical problems and statistical problems or challenges for single cell data analysis. I've already referred to a couple of times into sort of various things that can introduce variation in gene expression levels. And understanding the sort of how different technical and biological factors can give rise to differences in gene expression patterns is one of the key things in being able to analyze transcriptome or actually any type of omics data. So the kinds of things that we're talking about here can be sometimes very big things that kind of like actually shift the expression levels of like maybe even thousands of genes. Sometimes they can be more subtle. And when we talk about technical factors, we can refer to things like, for example, RNA extraction that can affect RNA quality and quantity, and those kinds of things actually affect the data that you end up getting out in the end. The process of library preparation and sequencing, there can be subtle technical differences there, and then also properties of sequencing data. So for example, how many reads did you end up getting from each particular sample? There's typically at least small differences there, and you want to kind of make sure that doesn't affect your analysis because it's somewhat arbitrary thing. And these things can vary both in terms of sort of like actual batches. So like you extracted one set of samples today and another set of samples next week, and there can be differences between those groups. But there is also kind of like just variation between samples. Even if they're part of the same batch, there's always some degree of technical factors. And then when it comes to biological variation, there is, of course, I mean, let's say you're studying you collected monocytes from 100 individuals. There's, of course, variation in age and sex and this and that, all the kind of like the whole spectrum of variation between individuals. And even in, let's say, an inbred mice population, there are inter-individual differences. And that's something that can introduce variance in the data. And a particularly important aspect here is cell type composition if you're analyzing bulk tissue data. So if you're not, like you haven't exactly extracted monocytes, but you took blood samples from 100 individuals, and then you have extracted RNA from blood and done gene expression analysis with RNA sequencing, what you basically end up having is that, different individuals will have different proportions of different cell types. And because different cell types have different gene expression levels, that variation manifests very strongly in the differences that you see between different individuals. And this is something that really needs to be taken into account in the analysis. And sometimes those differences can be kind of biological, that let's say someone's leukocyte levels are high because they actually just had an infection. And that's sort of like you're in some ways capturing real biological variation between individuals there. But sometimes it can also be technical. For example, if you're analyzing a solid tissue that was cut out by a surgeon, how they happen to cut let's say a piece of nerve, how much of the surrounding tissue did they end up getting, then that's more like technical. So sometimes the difference between what is a technical factor and what is a biological factor is not always super clear. And some of the sources or some of these kinds of factors will be known to you, and that's often what we call metadata, where you try to kind of record all the kind of like properties of the subjects and samples and technical processing steps. When did you extract the RNA, and what was the how many sequencing reads did you get, etc. But some of these factors may be unknown. And you would then still typically kind of like, you need to keep in mind that there may be unknown sources of variance. Just to give you a little sample, so this is an old paper of mine now, 10 years old, but this is one of the first large RNA sequencing data sets of very clean data from cell lines. So this is kind of like cell lines grown on the dish, single cell type, very kind of sort of pretty much the sort of like an experiment that minimizes technical variation, and you still have quite a bit of gene expression variance explained by, for example, RNA extraction batch, library preparation date, primary index, sequencing sort of lanes, etc. The residual variation here, like that is the inter-individual variation, that like all the biology is somewhere here, but there is considerable technical variation here, and it is very important to understand that technical variation is a fact of life. It is always going to be there, and you need to design your studies so that it is robust to these effects. When we talk about human tissue data, that has much more sort of complexities, including cell type, composition, etc. And this is post-mortem tissue data from GTEx. You can see that, and this is multi-tissue analysis. So you start to have all kinds of like ischemic time of the donor, like how long they were dead before the sample was harvested, and different kinds of things starting to affect the variance in these data. But so that sounds a little bit depressing, right? But it's fine. There are ways to deal with these kinds of covariates, and actually in many ways what I'm saying here is true to not just to transcriptomics, but all kinds of large scale omics data. And I know that there is also other lectures on different data types. So many of these things apply to that as well. So the most important thing is to kind of like make sure that your biological factors of interest are not confounded with technical factors. So if you want to compare gene expression between cases and controls, don't extract RNA from the cases first and then of the controls. That's bad study design. You would obviously want to follow good laboratory practices to reduce technical variation in the data. There is always going to be some, but you can reduce it. It's always a good practice to include biological and technical replicates to be able to assess the sources of variance and kind of like understand what's going on. And then, as I mentioned before, there's sort of the exploration of your data, of just kind of like making a PCA plot, etc., and seeing like what does PC1 correlate with, what's the clusters that we see in PC2, etc., trying to understand what's actually contributing to variance in the data, what are the major and minor drivers of gene expression differences, is a really key step. The nice thing is that even though, so technical sources of variation is these are present in all molecular data. Even if you do a Western blot or a qPCR or any kind of a molecular analysis, you will always have some technical differences there. But what is nice about large-scale omics data, like transcriptome analysis with RNA sequencing, is that you can actually see these patterns. You can detect them. And this is kind of like one of those things that sometimes people think that, let's say, RNA sequencing is not as robust, or it's kind of like that there's always all these technical factors. I mean, the technical factors are there in the, let's say, qPCR data from like 3 genes as well. It's just that you don't know about it. And the fact that the large scale data that we have, its depth, its diversity, allows us to detect what are these sort of different factors contributing to variance, makes omics data better and not worse than the traditional small scale data set. It allows us to make more robust discoveries. But the important thing here is to think very carefully, what's the biological effect of interest? Like, what are you actually studying? What's the question? What are the other kind of things that can give rise to those patterns that you're looking for, and how you can sort of remove those things? And that's basically kind of like the process of doing science and designing your studies, which is what we all like to do. I'll finish just with a couple of notes about a few mentions of few very key transcriptome data or method resources. So I've already mentioned Genotype Tissue Expression Project, or GTEx. This is also where Casey was involved in the sort of earlier phases of GTEx. And GTEx provides very, very sort of deep analysis of transcriptome across postmortem human tissues, also some single cell data. This is just an example. Like, you can basically take your gene of interest and see how it's expressed in different tissues, and looking at I mean, gene expression levels, and also kind of single cell exon expression, splicing patterns, etc. And a particular feature of GTEx is that the main point of that project was to map genetic regulatory variants. And I know that you'll be talking about EQTLs, etc. later, but GTEx is a key resource for those types of data, but also just basic sort of transcriptome characterization. Human Cell Atlas, that just released its huge batch of papers, is a key resource for single cell atlas data, so characterizing transcriptome at the single cell level and across different cell types of human tissues. And ENCODE is a great resource, particularly for, I mean, they do have tissue data as well, but for example, cell line data, etc. I mean, some of the experimental models that we use, and also non-human samples. And a particular strength of ENCODE is that of course they have not just, I mean, they do have very good transcriptome data, including long read transcriptome data, but they also have other omics data, and a lot of it. I want to finish with a little bit of a sort of a few points about what I see as the sort of ongoing and upcoming focus areas in transcriptomics. This is at a very, very high level, but it's important to think about the big questions, like what are we actually trying to do here now? And of course, a key question is how does the genome actually work to regulate gene expression, transcription, etc.? Trying to understand the regulatory code of the genome and its dynamic nature. And here, the sort of the key way to make progress is you can't really solve this question by staring at the transcriptome only. I mean, this is fundamentally a little bit like a genomics question. So you need to also have data of the genome, its variation, and the epigenome, and the various layers of that. And I know that there will be many later lectures discussing these aspects, as this was some of the kind of key questions that also Casey asked in his research program, and me as well. But when we think about a little bit more in the sort of like not so much genetics questions, but kind of like moving on to the sort of questions like how do genes drive cellular function, thinking about those regulatory networks, how do genes regulate each other, so we have gene expression and kind of like then what, that type of a question. There it becomes important to integrate transcriptome data with, for example, proteome data, and of course a lot of sort of sophisticated statistical models, and also genetics and CRISPR type of perturbations of the system play a role here. There is also an increasing amount of transcriptomics and other sort of functional omics applications in precision medicine, thinking about molecular markers of disease. So this is a little bit less about sort of like understanding sort of like molecular biology, but more just kind of like, can you for example, measure gene expression in people and then make some sort of better diagnosis or better estimates of progression of disease or disease state. And here it's important to kind of integrate transcriptome data and whatever other omic data you have, often genomes, etc., with different clinical data sets. And this is like a whole sort of area of its own, very, very interesting and a growing one. And in terms of the key technologies that the field is focusing on now, at least sort of like here focusing specifically on the transcriptome space, there is the obvious growth of single cell technology, scaling things up, diversifying the methods, making further applications, more multi-omics at single cell level, etc. Long-read transcriptomics is very, very important. Much of the field has focused on analyzing gene expression when actually the true functional molecules of the transcriptome are transcripts, not genes. And now with long-read transcriptomics, we have an increasing ability to capture those molecules and characterize them. And I think that this is going to be revolutionary for transcriptomics. It's been a revolution that is been a little bit sort of like slowly happening, but I think that there can be some real breakthroughs here. And always when we talk about analysis of biological questions from these large and complex data sets, statistical methods development, and I should also include in that the kind of bioinformatics tool development is extremely important to actually extract biology of complex data. And the transcriptome field is at this kind of like a nice intersection where it is informative of both sort of like deep molecular mechanisms, but also the kind of applications in the areas of molecular precision medicine. And I think that making progress in both, I mean, these two questions are very much married to each other and interact in interesting ways. But I will end here. Thank you all very much for listening. And I hope you enjoy the rest of the lectures in this series as well. Bye for now.